Hello friends, we are discussing transduction in bacteria. In the first part, we have already discussed about generalized transduction. In this video, we are going to discuss about specialized transduction. So first of all, definition. It is the process in which bacteriophage virus transfer only specific region of host, that is donor genome, to recipient bacteria. In one line or in one sentence, it means transfer of specific donor genes, and it is carried by temperate phage. Specialized transducing phages are formed only when phages from lysogenic cycle enters lytic cycle and phage progeny are released. Now let us see the steps or mechanism involved in specialized transduction. Temperate bacteriophage, when infects bacteria and injects its genetic material, then during lysogenic cycle, temperate phage DNA integrates into the host genome. Then phage DNA is called prophage when it is induced in the presence of ultraviolet rays or certain chemicals. Then temperate phage initiates lytic cycle. In this process, phage DNA gets excised from host genome. Sometimes what happens, imprecise or incorrect excision occurs in this process. Hence, excised DNA now includes bacterial genes lying adjacent to the prophage. Then new viral particles that are packed with viral as well as part of bacterial DNA are released. During infection to the another bacteria known as recipient bacteria, it injects its genetic material. Hence, donor bacterial genes are transferred to recipient bacteria. These donor genes can be inserted into the recipient chromosome or remain in the cytoplasm. The recipient bacteria then displays the newly acquired genetic characteristics. Now let us understand the specialized transduction with the help of diagram. When temperate bacteriophage infects bacterial cell, it injects its genetic material in the bacterial cell. This is the donor bacterial cell. During the lysogenic cycle, this phage DNA or genetic material is integrated with the bacterial genome. Now the phage DNA is known as prophage. When it is induced in the presence of certain chemicals or exposure to the ultraviolet radiations, then this prophage is excised from the bacterial genome. Sometimes the excision is imprecise or it is incorrect, as a result of which some of the genes that are present adjacent to the attachment site of his phage DNA is excised along with the phage DNA, and some of the phage genes are lost. When this enters the lytic cycle, new phage particles are formed which consist of part of phage DNA and part of bacterial genes. Now these are known as transducing phages. Then these are released by the lysis of the bacterial cell. When this transducing phage infects another bacterial cell known as the recipient bacteria, then it injects its genetic material in this bacterial cell. After this, either only the donor bacterial genes or both the phage DNA and the donor bacterial DNA is integrated in the recipient bacterial genome. And then this recipient bacteria displays the genetic characteristics. This is known as specialized transduction. Now we may think that why the term specific transduction is used. Specific transduction is used because the genes that are transduced are specific. Those genes that are near the site at which prophage is integrated. That means these include only the neighboring ones. And hence, in this process, restricted set of bacterial genes are transferred to the another bacterium, not random genes. Hence, the term specific transduction or specialized transduction is used. One of the example of this type of transduction is phage lambda and E.Coli. Here phage lambda is temperate phage and E.Coli is the bacteria. Now let us understand the mechanism of transduction for phage lambda and E.Coli. So what happens, as we have seen in the steps of specialized transduction, phage lambda infects E.Coli and injects its genetic material, which is then integrated between the gal and biogenes of E.Coli genome. It remains integrated for several generations and is replicated along with the bacterial genome. When it is induced in the presence of ultraviolet radiations or certain chemicals, then these phage genome or phage DNA, which is known as prophage, are excised. In most of the cases, normal excision takes place, as a result of which new phage contains no bacterial genes and it is complete, because the entire phage DNA is excised from the bacterial genome. In rare cases, abnormal excision occurs, and this occurs due to certain errors. So in this imprecise or abnormal excision, what happens? Gal or biogenes which are present adjacent to the attachment site of prophage or phage DNA are taken up, and also some of the phage genes are lost. Hence the lambda phage, that is transducing phages that are created, are defective. These carry bacterial genes and also part of phage genome, and some of the phage genes are lacking, and these are known as rare recombinants, as this type of the excision is rare. So when such transducing phage particles are released and infect another bacterium, that is recipient bacterium, these can transfer them to that bacterium. It means specific genes of donor bacterium are transferred to the recipient bacterium by phage lambda transducing particles. So it is the example of specialized transduction. Now let us understand mechanism of transduction for phage lambda and E.Coli with the help of this diagram. When the phage lambda infects the bacteria, in this case E.Coli, then it injects its genetic material in the host bacteria. Since it is temperate phage, so it will undergo lysogenic cycle, as a result of which this phage lambda DNA is integrated in the bacterial DNA. So this is the attachment site for the phage lambda DNA. It gets integrated in the bacterial DNA. This is the integrated phage lambda DNA, and adjacent to this integrated phage lambda DNA are the gal and biogenes of the bacteria or the host bacteria. So it remains integrated for several generations and replicates along with the bacterial DNA. When, due to certain induction like presence of any chemical or ultraviolet radiation, lytic cycle is induced. In this case, this integrated phage lambda DNA is excised. So the excision of phage lambda DNA takes place. In most of the cases, the excision is normal. So when the excision is correct or precise, then new phage particle contains only phage lambda DNA. No bacterial genes are present, hence it is complete. New phage particles are produced. Then the lysis of the bacterial cell takes place and these are released. But sometimes what happens? The excision is incorrect or improper. This occurs rarely. In this imprecise excision, some of the bacterial genes are taken by the excised phage DNA. In this case, gal or biogenes are taken up, which is adjacent to the integrated site of phage lambda DNA. And some of the phage genes are lost it remains integrated with the bacterial DNA. Hence, defective lambda phage is created, which carries bacterial genes and part of phage genes. So, when new virus particles are formed, these are known as transducing phage particles. These are released by the lysis of the host cell and then transfer the donor bacterial genes to the new bacterium, that is known as the recipient bacterium. So, here transduction occurs by the phage lambda DNA, that is, the donor bacterial genes are transferred to the recipient bacteria. Hence, it is known as the transducing phage particle. Here the gal genes are for galactose fermentation and bio genes are for biotin synthesis. Now let us see the rare recombinants. First, lambda gal particles. These lack tail genes and sometimes head genes, and these are located at the right end of the prophase, that is phage lambda DNA. Since these are essential genes, hence lambda gal transducing particles cannot perform lytic cycle. That is, these cannot reproduce in the recipient bacteria. Therefore, these are defective and hence these are denoted as lambda D gal, where D stands for defective. So, what we have seen that transducing particles lack tail and sometimes head genes. So, now you might be thinking that how these transducing particles are formed. So, initially when these are formed in the donor cell or the donor bacteria, there is no problem, as prophase, which is integrated in the bacterial DNA, contains all the essential genes. This prophase only undergoes aberrant excision, and the part of phage DNA which is lacking is present integrated with the bacterial genome, as a result of which necessary gene products, that is head and tail proteins, are still present in the host cell, and hence transducing particles are formed. But once transducing phages are formed and infects new cell, then there is deficiency of these essential gene products, as these lack head and sometimes tail genes. That is, certain essential genes are lacking, so cannot reproduce independently or lytic cycle do not take place. Second is lambda bio. In this, transducing particles lack genes from left end of the prophase, that is INT and XIS. Hence, phages are usually plaque forming called lambda P bio, as missing genes are not essential for lytic growth as in lambda D bio, where head and tail genes are missing. Here P stands for plaque forming. Now let us understand these rare recombinants with the help of the diagram. So transducing particles, as we have seen, are formed when the imprecise or incorrect excision of the prophase or phage lambda DNA takes place. And since it occurs rarely, so the recombinants that are produced are known as rare recombinants. There are two types of the rare recombinants. Sometimes what happens, the imprecise or incorrect excision is such that tail genes or sometimes head genes are missing or these are lacking during the excision, and the gal bacterial gene adjacent to the integration site of the phage lambda DNA is excised. Then transducing particles that are formed are known as lambda D gal, where D stands for defective. So here essential genes which form the head and the tail proteins are missing. Hence these are defective. In the donor cell, yet transducing particles are formed because these proteins are already present in the donor cell, as these genes are present integrated in the donor bacterial DNA. But when these transducing particles are released by the lysis of the donor cell and infect any recipient bacteria, in that case, these transfer the donor bacterial gene to to the recipient bacteria. So here they are performing specialized transduction, but new viral particles are not formed because here the essential genes, head and the tail genes, are missing. Hence, essential gene products or head and tail proteins are missing. So, new viral particles are not formed. That means virus is not able to reproduce or perform lytic cycle. In some cases, excision is such that genes that are present to the left end of the prophase or phase lambda DNA is missing. And biogenes that is present adjacent to the right end of the integration site is included. This biogene is the gene of bacterial DNA. So, in this case, the transducing phase particles that are formed are known as Lambda P bio. These transducing particles are released by the lysis of the donor bacterial cell and then infects recipient bacteria. These are able to reproduce or these perform the lytic cycle because here the essential genes are not lacking. Head and tail genes are present. The genes which are lacking or missing are non-essential. So, new viral particles are produced and then these are released by the lysis of the recipient cell. These are known as Lambda P bio, where P stands for plaque forming. Because of the lysis of the recipient bacteria, a clear area is produced on the opaque field of the bacteria, which indicates that certain bacterial cells are lysed. And these clear areas are known as plaque. That's why these transducing phase particles are known as Lambda P bio, where P stands for plaque. This is all for today's video. If you like this video, please hit the like button, share it and subscribe my channel. Thank you.